Hey there, when you're designing an experiment, you always have two fundamental choices to make that experiment more complete or more comprehensive. And this is what we're talking about now. This is very important to know about because it tells you how you can design basically a useful study and a more complete study. Why would you want a more complete study, a more comprehensive study? Well, it may just satisfy your scientific curiosity. You may need a more complete study, for example, to complete a full thesis chapter or to fulfill project goals. This may also be the difference between a short communication and a full paper, for example. And here are the two different ways to make an experiment more complete. You can either modify the treatment structure and make that more complex, or given a certain treatment structure, you can make an experiment more complete by measuring more things in the end. Typically, you can't do both because the effort just becomes unmanageable in many cases. So now let's go over these two ways of making your experiment more complete. First, treatment structure. This typically means including other factors, other settings, other parameters, and other players in your design. Typically, maybe in the form of factorial experiments where you have one factor crossed with another factor. For example, warming and drought in all combinations of warming and drought. Now, one thing to remember is when you do this and when you keep adding factors, parameters and settings basically, or players to your experimental design, the number of treatments tends to increase pretty quickly. And this is something to watch out for because it makes an experiment more difficult to manage, and this can quickly get out of hand when you have too many factors involved. The advantage of doing this, of adding more cases or settings or factors, as you want to call them, into your experiment, is that you are achieving greater external validity, meaning your experimental results can be generalized to a broader parameter space. Like, for example, if you're doing an experiment in forest and you're also doing this experiment in grassland as in another setting, then you might be able to make a more general statement. Or if you're doing an experiment under drought conditions and well-watered conditions, then you can also make a statement that encompasses these conditions, and so on and so forth. So basically it means when you have different soil types, different plant types or whatever included in your experiment, then this circumscribes basically the space over which you can generalize your experimental results. And that's highly desirable. So not only does it allow you to identify under what conditions your particular result still holds up, but it may also identify very important parameters or variables really that influence that outcome. Like, for example, a certain pollutant may be much more important under drought than under well-watered conditions. And if this is the case, this is of course super important to know. Now, the bottom line with this is like this can always be made more complicated. This is basically endless, right? So, I mean, the thing to think about in your experiment, what is the most important factor that you really want to make a statement about? Because you cannot make an experiment endlessly complicated, not only for logistical reasons, but also because it becomes much more difficult in terms of statistical power to detect any interactions of higher order. And they're also super difficult to interpret. So make sure you limit the set of factors that you include in an experiment so that you can in the end really still interpret the results. So the choice of these factors should really be based on scientific need. So this is about the treatment structure, and you can clearly see how that can make an experiment more complex and more complete. So what about the response variables? This means you are measuring a greater number of parameters or response variables at the end of your experiment or in between. And this can mean measuring the same thing, like microbial community, for example, at different levels of resolution, from high throughput sequencing to just measuring microbial biomass or enzymatic activities or the abundance of larger groups, to metagenomics or whatever. So you can basically increase the resolution at the end of your response variables. Increasing the level of resolution or also learning about what other parameters may have been affected in your experiment, of course, helps you figuring out what may have been going on and basically to get closer to identifying the chain of causality that was in effect in your experiment. I mean, you obviously applied a treatment, let's say drought, and then you saw a response, but you're not really sure how that response really unfolded unless you have measured multiple response variables, so you can reconstruct the chain of causality, as it were. And so having more response variables added that gets at that will be helpful in figuring out what has actually been going on in your experiment. As you're looking at responses in greater detail, you can also identify responses that otherwise may have been hidden. For example, if you look at responses at a relatively more coarse level, like let's say plant biomass, you may have overlooked responses like at plant community composition level. And so if you're looking at responses in greater detail, which implies more effort, you can uncover responses that you wouldn't otherwise have seen. But of course, don't measure everything out of the book just because you can. What you measure should be, of course, determined by scientific need and your question. And this part with the response variables is important to know because this, of course, means you can make the same relatively simple experiment with a relatively simple treatment structure, for example, more complete by just increasing the resolution of what you measure. That's very important to keep in mind as you plan experiments. Now, what does this all mean? Well, it means that this is important to keep in mind as you plan your experiments. You can basically have two screws in which you increase your level of completeness in an experiment. You can make from the very beginning your treatments more complex to reflect the greater parameter space of reality or important factors that may impinge on your results. This is, of course, you need to do that from the very beginning because then the experiment is set up like that. The other part, the response variables, you are slightly more flexible. Of course, you should also have in mind what response variables you're going to measure for your experiment. Obviously, you always have that. But as you see more interesting results, maybe you can also afterwards increase the resolution. It is also typically an either or. So you cannot have a very complicated treatment structure and then expect to also measure a huge list of response variables, right? Because that simply can get out of hand very quickly. So you have to think about when I have like an experiment with hundreds or maybe even thousands of experimental units, then I should really plan on measuring something pretty simple because otherwise this is not manageable. On the other hand, if you have a relatively simple design with only few treatments, then it is much more reasonable to think from the very beginning about measuring a larger number of response variables. So there's room for both of these options and you should be actively including them in your planning for your experiments. So thanks for watching. Let me know what you think in the comments. I'll see you in the next video. Bye.